Everybody loves this part where you get to wave at yourselves for a minute. So exciting. All right, so we're going to try and move expeditiously through this lesson. So let's try and all have really good focus so that when I ask a question, we get an answer and we can just roll right through it. Okay? Coolness. Let's go. So first, we're going to talk about RNA. So the role of RNA, like DNA, it's a nucleic acid that consists of a long chain of nucleotides, but how does it differ? There are three important differences. The first is that the sugar in RNA is, Eunice? Oh, okay. Who does mean to raise their hand on this one? Okay, Lucas. Yes, it is ribose instead of deoxyribose. So it's ribonucleic acid, not deoxyribonucleic acid. Second is that it is generally what kind of a strand, Kenneth? Single strand instead of double strand, like DNA. All right, and then it contains what in the place of thymine? This time, Eunice. There you go, uracil. So where you typically have thymine in your DNA complementary strand, in RNA, we don't have thymine, we have uracil. So if you see a chain of nucleotides and there's a U, you know you're dealing with RNA. If there's T's, you know you're dealing with DNA. Okay, so what are the functions of RNA? You can think of RNA molecule as a disposable copy of a segment of DNA, or like a working copy of a gene. So RNA molecules are involved in what kind of synthesis? What are we making with RNA? Caitlin? There you go, in protein synthesis only. So the whole purpose for RNA is to make proteins. RNA controls the assembly of what into proteins? What make up proteins? The building blocks of proteins are, Colin, amino acids. So you get a bunch of amino acids. They hook together and make this big, long chain. The bonds are called peptide bonds, so the chain is called a polypeptide chain, right? And when you get this big chain, it folds itself up and it becomes your protein. So your polypeptide chain, all the little amino acids are building blocks of the protein itself. And we have three main types of RNA. We have mRNA, which is messenger RNA. And what's he doing? What's he do?
He carries copies of instructions for polypeptide synthesis from the
Yes, he carries the information from DNA to the other parts of the cell. mRNA is made where? Think about it. It's made from DNA, so it's made where? In what part of the cell? Where's DNA? In the nucleus. Okay, our mRNA is made in the nucleus because DNA exists in the nucleus. And then our mRNA can leave the nucleus, go to the cytoplasm, and try and find ribosomal RNA, or a ribosome, which is rRNA. And what's the purpose of ribosomal RNA, Caitlin?
It forms an important part of subunits of ribosome.
Yes, so rRNA is what makes up the ribosome. So you have DNA in the nucleus. It can be transcribed into mRNA, which can leave the nucleus to a ribosome, which is made of rRNA. So now your mRNA is in your ribosome made of rRNA. And the last type of RNA is transfer RNA. And what does it do? Oh, Xander. Yes, it carries amino acids. When the protein is built, the transfer RNA molecules transfer each amino acid to the ribosome. Okay, important fact. We need amino acids to live. Some of them we can synthesize ourselves. Others we cannot. Where do we get them? This is why you have to eat protein. This is why protein is very important, because protein breaks down into amino acids. There are some people, some disorders that cause people's bodies to treat certain proteins as invaders, and their immune system attacks them when they eat them. It's not like a typical allergy. Some of those people can only survive by drinking or using a tube, a stomach tube, to be fed particular type of formula that is only amino acids. They've taken the proteins and they've broken them down. So it's like pre-digested, because we need those amino acids to function. All right, so how does the cell make RNA? In transcription, segments of DNA serve as templates to produce complementary RNA molecules. The base sequences of transcribed RNA complement the base sequences of template DNA. It's not complement. It's not, oh, you look great today, I'm giving you a compliment. It's a complement, meaning it is the pair, right? It is the opposite side. It is the piece that matches with it. And transcription, Think about a scribe as someone who writes or codes, right? Transcription is the process of taking DNA, turning it into RNA. So RNA synthesis, transcription in prokaryotes, RNA synthesis and protein synthesis take place where, Eunice?
Cytoplasm.
Why?
Because
It's a prokaryote
there's no nucleus.
because it has no nucleus. So it has to be in the cytoplasm. Exactly. In eukaryotes, RNA is going to be produced where? Xander? Yeah, why in the nucleus? That's where the DNA is. It has to be made from the DNA, and the DNA is in the nucleus. Okay, so in transcription, it requires an enzyme. Anybody know what this enzyme is called? Yes. RNA polymerase, which is similar to DNA polymerase. Okay, so this little enzyme is necessary. Remember, enzymes are things that speed up chemical reactions. So you know they lower the activation energy. So you know there's something going on here. There's something going on in here that is involving some degree of chemical reaction. So promoters. All right, we have these things called promoters. RNA polymerase is going to bind only to, what'd you get, Lucas? Promoters. And these are regions of DNA that have specific base sequences. So RNA is not just going to like randomly bind anywhere. It's going to look for a specific sequence, like the ones that say start, for example. And then there's editing. So this is what we talked about that I said is a little more complex. So RNA editing, portions that are cut out and discarded, what do we call them, Caitlin? Yeah, we call those introns. These are portions we're going to get rid of. See them right here? Chop, chop. We don't care about them. The remaining pieces are called, oh, Ridley. Exxons, not like the gas station that used to be everywhere. We don't see too many Exxon gas stations anymore. Exxon had, interestingly, Exxon had a promotion for a while where they had this little plush tiger tail because their tagline was like, put the tiger in your tank, like fill up your car with our gas and your car will perform well, put the tiger in your tank. And they had these little plush tiger tails that people left hanging out of their gas cap, like dangling on the side of their car for a while in the mid to late 80s. We had one. I stapled it to the back of my dad's jeans and he wore them to work that way. So we had a prank thing going where we would trade off who pranked each other for several years, and it went on forever. It went so far. The one thing we pranked each other with was this. It was this little eyeball that when you rolled, it always faced up. You know, you ever seen those, like a little plastic thing, and you roll it and always faced up? He went so far as to mail it to me. I was so excited because I got a package and it was just the eyeball. It was like the biggest punk of my life. I'm like, I thought I got a treat.
Start with you. They support your entire.
Okay, that's a long one.
With Thrivent, your money can be a force for good for the people, causes,
Yay.
and things you're most thankful for. Thrivent, where
All
money
right.
means more.
They're all banking commercials these days. It's better than political.
Sometimes it feels like DNA gets all the credit. Yes, the structure of DNA is amazing. It has a beautiful double helix structure, a twisted ladder. It has its own emoji. DNA stores genetic information and codes for your traits. However, sometimes what gets left out is how important RNA is. Without RNA, you actually couldn't get that genetic message out to your cells so that they can start producing protein. We talk about this in protein synthesis. RNA is a very important biomolecule, just as important as DNA. In fact, RNA is even hypothesized as coming first, before DNA, in the RNA world hypothesis, something we need to make a separate video about. So let's compare and contrast DNA with RNA. First, you will find DNA and RNA in all living organisms. In eukaryotic cells, DNA tends to be found in the nucleus, while you can find RNA both inside and outside of the nucleus. Prokaryotic cells don't have a nucleus. Both DNA and RNA are nucleic acids, which are a type of biomolecule. Nucleic acids have a monomer, which, if you remember from our biomolecules video, a monomer is a building block. The monomer for nucleic acid is a nucleotide, so both DNA and RNA have nucleotides. The nucleotides of both RNA and DNA have three parts: a phosphate, sugar, and a base. Let's draw 6 nucleotides of DNA and 6 nucleotides of RNA. DNA is generally double-stranded, and if focusing on these two strands of nucleotides here, you can see they run anti-parallel to each other. RNA is generally single-stranded, so you are just seeing one strand here. The sugar in DNA is deoxyribose, and the sugar in RNA is ribose. This makes sense because DNA stands for deoxyribonucleic acid. That's helpful to know because the deoxyribose is a sugar, and nucleic acid is that type of biomolecule it is. RNA stands for ribonucleic acid, as its sugar is ribose. The bases in DNA are adenine, thymine, guanine, and cytosine. It helps to remember the popular mnemonic apples in the tree. That helps you remember that A goes with T, and car in the garage. So C goes with G, to understand how DNA bases pair. The bases in RNA are adenine, uracil, guanine, and cytosine. Notice the different one? It's uracil. So you have to change that popular mnemonic device here. Instead of apples in the tree, maybe the apples are under. Car in the garage still works though. We mentioned earlier that DNA codes for your traits, but it couldn't do that without RNA's help. In our protein synthesis video, we talk about 3 different types of RNA and their very important roles. mRNA, which stands for messenger RNA, mRNA's job is to carry a message based off of the DNA. In eukaryotic cells, DNA generally stays in the nucleus, but mRNA has the ability to leave the nucleus to take the message to a ribosome. Ribosomes make protein, and RNA is actually a major component of ribosomes. This type of RNA is called rRNA, which stands for ribosomal RNA. Finally, we discussed transfer RNA, more abbreviated tRNA. Its job is to transfer amino acids to match the correct mRNA codon. Codon charts using mRNA codons have been developed so that you can actually see which amino acid is brought for each mRNA codon. When those amino acids are joined together, they make a polypeptide chain. Proteins are made of one or more of these polypeptide chains, and proteins have tons of different roles, but we don't want to spoil it. Check out more in our protein synthesis video. Before we go, let's try a little 3-question quiz, shall we? Just pause it after the question so you have time to think about it. Question 1. If I have 8 DNA nucleotides, how many DNA bases do I have?
How many bases would you have if you had 8 nucleotides? Well, you've got 1 here. Yep. So if they're in pairs, so how many bases do you have for 8 nucleotides? You have, here's a nucleotide, here's a base. 8. And then if they were in pairs, you'd have.
And also, how many base
pairs you'd have 16.
The answer.
Right.
Each nucleotide, regardless of whether it's a DNA or RNA nucleotide, has a phosphate, sugar, and a base. So 8 DNA nucleotides would have 8 bases. DNA bases pair like this, and that's 4 DNA base pairs.
4 pairs.
Question
Sorry. Yeah.
2.
Not
If
no,
one strand
8
of
bases
DNA
is 4
has
pairs.
these
Not
bases
16.
shown here,
Yeah. My brain died.
ATTGAZ. Can you complete what the complementary DNA bases would be for the other DNA strand?
Hey, everybody together. It's T, A, C, C,
G.
T, G. Right. Okay.
The answer. So remember those base pairing rules for DNA and the popular mnemonic. If placing the bases in this image, reading in the direction shown on this image, they'd be TAACTG. Question 3. In a process known as transcription, a complementary RNA strand called messenger RNA has to complement the DNA. More about that in our protein synthesis video. So if I still had the original portion of DNA here, what would be the complementary RNA bases?
What would they be?
U,
U.
C, U, G.
You guys have got this.
The answer.
Love it.
So remember, it's asking for RNA. No thymine here, it's uracil instead. Apples under helps remember A with E.
You've got it. All right. So let's have a look at this. So your answers are going to be DNA, RNA, or both. I'm a nucleic acid. What's the answer, everybody? Both. All right. I'm usually single-stranded. What's the answer? RNA. I'm generally found both inside and outside the nucleus in eukaryotic cells. Who says RNA? Who says DNA? Who says both? RNA, because remember DNA lives in the nucleus. RNA can go in and out. mRNA can. I'm arranged as a double helix. My shape is described as a twisted ladder. DNA. I include bases guanine, cytosine, and adenine.
RNA.
Both. Each of my nucleotides includes a phosphate, a sugar, and a base. Both. I include the base uracil. RNA. I include the base thymine. DNA. I generally remain in the nucleus in eukaryotic cells. DNA. I have the sugar deoxyribose. And I have, wait a minute, I am made up of nucleotides. Both. And I have the sugar ribose. RNA. Good job. Excellent. Okay, so ribosomes and protein synthesis. How are they making proteins? The genetic code is read 3 letters at a time. So each word is 3 bases long, and it corresponds to a single what? What is it, Caitlin?
Codon.
Yeah, the codons correspond to a single what? Each codon equals a, Ridley? There you go, amino acid. Yes, so each codon equals an amino acid. Proteins are made by joining amino acids together into long chains, and those are called what, guys, everybody? Polypeptides or polypeptide chains. Here is an example. These are all amino acids down here at the bottom, and you can see how they link together chemically to form your polypeptide chain. Excellent. These bases form a language or genetic code with just 4 letters, A, C, G, and U. And we did this in lab, and you figured out that if I went G, C, G, what amino acid am I going to make?
Alan.
Yeah, alanine. Very good. Yep. All right. Each 3-letter word, this is it, Caitlin, is known as a codon. Okay, each 3-letter word is a codon, right? Each 3 letters together make a codon. The methionine codon serves as initiation or start. What's methionine? Let's go back and look here. Methionine. Here it is, right here. So what is it? Very good. It's going to be AUG. That's methionine. That means start. Following the start codon, mRNA is read 3 bases at a time until it reaches one of 3 different stop codons. There are 3 different options for stop, only one for start. It is methionine, it is AUG. Yes.
Last year was the AUC stop.
No, that may have been one of the stops. Well, we'll see it in a minute, I think. But it shows you there are 3 options for stop. That may have been one of them, yeah. So translation, what role does the ribosome play in assembling proteins? Ribosomes use the sequence of codons and mRNA to assemble amino acids into, what is it again?
Polypeptide.
Yes, your polypeptide chain. So look here. In this case, your tRNA is shaped like a jelly bean, and it's adorable. I don't know why it has 3 legs, but it's so cute. I think the 3 legs are, yeah, oh yeah, here they are. Because they link down here to 3 little bases. That's very cute. So they come in, and they're each carrying, woo, their little amino acid, woo, and they bring it down, and they click on, and then the amino acids are going to bond together, and the little tRNA molecules will actually leave, and the little amino acid chain will continue forming. And that's how your coding happens. The decoding of an mRNA message into a protein is a process known as what? What you got? Yes. Okay. So, oh, sorry, I didn't even see you right there. You had a hand. So yes, translation. So what's it called when the DNA is being coded into mRNA? That one's called what? When the DNA is coded into RNA is transcription. And when RNA is coded into, mRNA is coded into proteins is what? You said it a second ago. The translation. Yeah, translation. Okay, there you go. So translation. There you are. So transcription happens where in eukaryotic cells? In the nucleus, because the DNA is in the nucleus. And translation happens in the ribosome and the cytoplasm. Right? There you go. So here are your steps. Your messenger RNA is transcribed in the nucleus and then enters where for translation? We just said it? Into the cytoplasm, into the ribosome within the cytoplasm. Yep, it leaves its little home and goes out into the world. Each tRNA, transfer RNA molecule, has 3 unpaired bases called the what? Anticodon. Very good. All right. The codon exists on your mRNA strand. The anticodon is what is brought in by the little tRNA guys. Remember the little guys holding their little balls? Woo! That they have their little feet, their 3 feet would be your anticodon, which is complementary to the mRNA codon. Remember this lady? Here's our friend again, Miss Sloan. This one's great.
Yeah. Hi, this is Miss Litton, and this is my wonderful period 3 class. Show them, show them. There they are. Yay. And we're gonna show you our song to remember transcription and translation. So we're starting with the DNA, it's a double helix, and we're going to what? Unwind and unzip. Helix, and since we're making RNA is single-stranded, so we have a leading anna, you will put the lagging strand away. And because we're making RNA, we're gonna use RNA polymerase binds to the promoter. We go boom, 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 then we're gonna make some mRNA. Now tell me about this mRNA. It has what sugar in it? Ribose instead of deoxyribose, and it has the base uracil instead of thymine, and it is what kind of stranded? Stranded. Okay, so RNA polymerase binds to the promoter. Boom, boom, boom. Make your mRNA. Peel it off. Go get your lagging strand. Zip it back up. Put your DNA away. Go get your mRNA. We're gonna put on a cap. A 7-methylguanosine and a tail. 100, 200 adenines, and then we're gonna cut out the introns before it leaves the nucleus.
Editing.
Then we're gonna take this mRNA, we're gonna go out to the workbench. I want to do the workbench. And every 3 bases on the mRNA is called a codon, which is complementary to a anticodon on what? tRNA. The T stands for transfer, and it's transferring an amino acid. tRNA 1 lands, tRNA 2 lands, the amino acids hook together, and we build up protein. Now, this part to this part was called what? A transcription. And where does that take place? In the nucleus, if you have one. This part here is called what? Translation. And the other song we learned, right? Right? Boom, 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 boom. That's called what? DNA what? Replication. And that's all part of the central
Dogma.
Good.
Yeah. There you go. So she says helicase, because that's what does the unzipping. If you remember, we briefly touched on that. But when the DNA is unzipped, the little enzyme that does the unzipping, she does a helicase because it's doing the unzipping. All right, so what's the central dogma she mentioned at the end of molecular biology? And it is that information is transferred from DNA to RNA to protein. What's a dogma? Anybody know what a dogma is, Drew?
Like the belief system of like a group of people, kind of.
Yeah, a belief system, a very specific belief system. So this is like basically, this is the heart of molecular biology, that information goes from DNA to RNA to protein. Central dogma. There you go. All right, so what happens, what is the way in which DNA, RNA, and proteins are involved in putting genetic information into action? Kenny, do you know? There you go. Okay, so yeah, gene expression is the way in which DNA, RNA, and proteins are involved in putting genetic information into action. DNA carries information for specifying what in an organism? What does DNA give us? Specifies our what? Our traits, right? All of our traits, except for those that we develop from socialization or lack thereof. Despite their enormous diversity in form and function, living, y'all are laughing, but it's true. Living organisms display remarkable unity at life's most basic level, the molecular biology of the gene. All right, here's their protein synthesis. And it's exactly what we just said. This is a review of what Miss Sloan just showed you, what we just talked about. Yeah, tools. All right. Right.
After learning about DNA, have you ever wondered how can the DNA actually result in traits? Let's take an example, like eye color. Yes, your DNA has the genetic information that codes for the color of your eyes. Your eye color is based on a pigment that is inside the eyes. But in order to have that pigment, you have genes, which are portions of DNA that can code for proteins, which help make that pigment. So what we're going to talk about is how your DNA can lead to the making of a protein. This process is called protein synthesis. Synthesis essentially means to make something. So protein synthesis means to make protein. And you may wonder, what's the big deal about proteins? Well, you may not realize this, but proteins are kind of a big deal. They do all kinds of things. Proteins are involved in transport, in structure, in acting as enzymes that make all kinds of materials, in protecting the body, and so much more. You've got to make proteins that's essential for you to live. And what is so cool is that you are making proteins right now as you sit and watch this video. It's happening in your cells. They're making proteins. So back to your DNA and its role in all of this. All of your cells have DNA, well, a few exceptions, and that DNA is in the nucleus. Some DNA is non-coding DNA. Some DNA makes up genes that are not activated. More about that in our gene regulation video, but we're going to talk about genes that are coding for active proteins. So how are we going to get the information from these genes out of the nucleus so that the cell can start producing the proteins that it needs to make? Well, let us introduce you to the amazing work of RNA. We have a video comparing and contrasting RNA and DNA. We'll be brief here in saying that RNA is a nucleic acid like DNA, but it has a few differences. Its role in protein synthesis also is huge. Before we get into the process, please note our typical disclaimer. We tend to simplify topics, but as always, we hope that you have the desire to explore this complex, amazing process later on, to learn all about the extra information that we don't have the ability to include in this short video. In protein synthesis, we can look at two major steps. One is transcription and the other is translation. Transcription has a C in it, and translation has an L in it. I remember that C comes before L in the alphabet, which helps me remember that transcription comes first. I like a lot of alphabet mnemonics. The transcription is when we're going to transcribe the DNA into a message. In your cells, the DNA is in the nucleus, so therefore we're doing transcription in the nucleus. In the step of transcription, an enzyme called RNA polymerase will connect complementary RNA bases to the DNA. These RNA bases are bonded together to form a single-stranded mRNA. The M in mRNA stands for messenger. Messenger RNA consists of a message made of RNA that has been based on the DNA. We do want to mention that this mRNA is not usually ready to go right away. There's usually a significant amount of mRNA editing that occurs. We highly encourage you to do some reading about that because it's not only fascinating, it's critical for the process to work correctly. So what's something great about being mRNA? Well, in eukaryotes, you get out of the nucleus. The mRNA can go out of the nucleus into the cytoplasm where it's going to attach to a ribosome. Ribosomes make protein. The ribosome is made of rRNA. And that's an easy one to remember because the R stands for ribosomal RNA. The ribosome is going to build our protein in the next step called translation. You know, you can find a lot of great clips and animations on translation that are just fantastic. We're just going to break down some basics of what's happening. In the cytoplasm, if you look at this, you have all these tRNA molecules available. tRNA stands for transfer RNA. They carry an amino acid on them. An amino acid is the monomer for a protein. It's a building block for a protein. Since we're making proteins, we're going to need those amino acids to build it. If you have a bunch of amino acids together, you can build a protein. So it's the tRNA that is going to bring those amino acids together to make that. But wait, how does the tRNA know which amino acids to bring? That's why the mRNA, the message, is so important because it's going to direct which tRNAs come in and therefore which amino acids are transferred. All of these tRNAs are looking for complementary bases. When they find the complementary bases on the mRNA, they transfer their amino acid. When tRNA is bringing in the amino acids, it reads the bases, represented by these letters here on the mRNA, in threes. So it doesn't read one letter at a time, it reads in triplets. That's called a codon. So for example, in this mRNA, the tRNA would read the codon AUG. One of these tRNAs contains a complementary anticodon, which in this case is UAC. All tRNAs that have the anticodon UAC will be carrying an amino acid called methionine. A tRNA with the UAC anticodon comes in to pair with the complementary AUG codon on the mRNA. It transfers the amino acid it carries, methionine. The tRNA will eventually leave, but it will leave behind its amino acid. That's the first amino acid before looking at the next codon. Before we do the next codon to carry this on, if you're wondering, how did you know that the tRNA that went with the AUG codon would be carrying an amino acid called methionine? Well, for that, you will find a codon chart.
So there are your stops that you asked
The
about.
codon chart to determine which amino acid each mRNA codon will code for.
UAA,
Isn't it so
UAG,
fascinating that scientists have been able to determine which
UG,
amino acid corresponds with these codons? I used to have a codon chart poster and just marvel at that. You can see on the codon chart that the AUG codon on the mRNA codes for methionine. AUG is also considered a start codon, as methionine is typically going to be your first amino acid in proteins. There are many types of amino acids in the codon chart, but there are even more possible codon combinations. That means there can be more than one codon that code for the same amino acid. For example, according to the mRNA codon chart, all of these mRNA codons here code for the same amino acid, leucine. That means their complementary tRNAs all carry the same amino acid, leucine. Okay, so going back to the mRNA, let's try the next codon on this mRNA, CCA. On the codon chart, you can see that codes for the amino acid proline. The complementary tRNA has the anticodon GGU. And look, there's the proline that we knew it would be carrying. The tRNA will transfer that amino acid and eventually leave where it can pick up another amino acid. These amino acids are held together by a peptide bond, and it will keep on growing. Typically, at the very end of the mRNA, there is a stop codon. Stop codons do not code for an amino acid, but when the ribosome reaches it, it indicates that the protein building is finished. So the result of translation is that you built a chain of amino acids that were brought in certain sequences based on the coding of the mRNA. But remember, that mRNA was complementary to the DNA. So the DNA ultimately was the director of the entire protein building. Of course, it couldn't have done it without some serious help from mRNA, rRNA, and tRNA. Protein folding.
So you get the idea. Yeah. And then they fold up, and then that's the shape that matters. So let's talk mutations for a bit. So what are mutations? Mutations are heritable, meaning you can inherit them. And what are they? They're what? Changes. They're heritable changes in genetic information. Mutations that involve changes in one or a few nucleotides are known as what kind of mutation? Do we know? You know? Point mutations. Very good. All right. And if a gene in one cell is altered, that alteration can be passed to every cell that develops from the original one. So if that alteration takes place in a germ cell, which is a cell that produces gametes, so cells that produce sperm, cells that produce eggs, then therefore you can get a new mutation, a new point mutation that's going to be passed only to offspring, but not affect the parent necessarily. And then you can also have alterations in your individual cells as they go through their lifespan. And then that can be passed to any offspring those cells produce as they divide and continue to produce copies of themselves. Point mutations include substitutions, insertions, and deletions. A substitution, one base is going to be changed to a different base. Insertions and deletions are point mutations in which one base is inserted or removed from a sequence. And insertions and deletions are also called frame shift mutations because they shift the reading frame for the genetic message. If you think about those three letters, they can shift the entire frame reading, and that can cause a real problem. So we're going to use this as our analogy. Obviously, we're not dealing with three letters, so this is not a particular codon. This is just a good word that works for analogy. Let's start with beast. I don't know what kind of a beast he is, a wildebeest, is something, a water buffalo. He's a beast, okay? And that would be your normal word, beast. Now, a substitution, we substitute the B for the F and we get beast. Okay, so now it doesn't mean the same thing anymore, right? It means something different, even though only one letter changed. Now, an insertion, we're going to add a letter. Now, let me tell you how hard it was to find this graphic appropriate for high school, not geared toward college, because I can tell you that is not the picture they give you for the college level. It looks a little more like your autopsy graphic that some of you saw today. So yes, breast. Now we've added an R to the word beast and we've gone from beast to breast. Okay, now how about if we have a deletion? Now we are taking out the A entirely. We've gone from beast to best. Now we're best. All right. Now what happens if we invert? Now beast is beats. So you can see how all of these slight changes can completely change the meaning of the word. And that will change how your coding is going as far as translation is concerned. And then you can end up with problems or not problems. It kind of depends on where the mutation is. So there are 4 types of, those are in your code. Those are code mutations, point mutations. But then there are chromosomal mutations. So you can have deletion, duplication, inversion, and translocation. So what involves the loss of all or part of a chromosome? Kenna? There you go. You got a deletion. Look at this. This guy's leg is like been chopped off over here. He's missing this entire part of this chromosome. And then what happens when you get an extra copy, Caitlin? Yes, an extra copy of all or part of a chromosome is a duplication. Right here, look. That's twice. It's got that code in there 2 times. All right. What is it when it reverses the direction?
Inversion.
That's an inversion. So if you look here, you see it's got like the big red band and then a big green band on the top. And over here, you got the big red band on the bottom and the green. So it literally turned itself upside down and translocated. And then what's this one? This last one is translocation, right? So a translocation, this occurs when part of one chromosome breaks off and attaches to another. Look, it's completely in the wrong place. Like it's completely attached itself to the wrong thing. All right, so what are the effects of mutations? How do mutations affect genes? Okay, Barrett, don't do that. Might be distracting, just so you know. All right, so the effects of mutations can vary widely. Many have little or no effect at all, and some produce beneficial variation. Some can negatively disrupt gene function. And we're going to talk about genetic disorders soon, and we're going to go over a whole bunch of them. So you'll see how some of these things can cause real problems. The cellular machinery that replicates DNA inserts an incorrect base roughly once every 10 million bases. So it happens all the time. Small changes in genes can accumulate over time, though, and then eventually affect an offspring, even if they didn't from the beginning. So then we have what are called mutagens, and mutagens are things that cause mutations. We call these epigenic because they cause mutations. So some of these are physical, some of these are chemical. Chemical mutagens include certain pesticides, a few natural plant alkaloids, tobacco smoke, environmental pollutants. So think about this, for example. We talked about what happens when cells divide, and don't get the message to die or stop growing and stop dividing. What's that called?
Yes.
Cancer. Okay, that's cancer. So if you look here, cigarette smoke is considered a mutagen. It can affect that part of the cell's genetic code that says, hey, dummy, it's time to die. Stop dividing and stop growing consistently, and then get that damage, then you can cause cancer. All right. You've also got physical mutagens that include some forms of electromagnetic radiation like X-rays and ultraviolet light. Did you know that when X-rays were brand new? Okay, who remembers if you were here for physical science, you might remember who was the man who invented the X-ray?
The guy with
The skeleton, huh?
the guy with he scanned his wife's ring. I forgot.
Yes, yes, there was an X-ray of his wife's ring. My skeleton model is named after him. Wilhelm Röntgen. Okay, Wilhelm Röntgen invented X-rays, and he did not patent it. He thought everybody should have access to it. I'm not going to patent this. This is really cool. He didn't really know what they were, so we called them X-rays. He just thought it was really neat. And people would build them in their homes for fun. and x-ray things for fun. That's great, right? And people have their own home-built x-rays. They can x-ray their hands like party trick. Look, I'm gonna show this. Okay, but the guys who ran the original x-ray machines, like when it became a thing, were like, oh, we're gonna start using these regularly to look inside of bodies. They calibrated the machine each morning by putting their hand underneath and getting an x-ray. And what do you suppose happened to their hands?
Cancel.
You can look up pictures of this. They have like, bones sticking out, just all the skin just gone, all the tissue just gone, hands just rotting away, practically pieces of them falling off. Oh yeah, it's real fun. You remember the Radium Girls? For those of you who know about the Radium Girls? Yeah, the girls that used to paint the faces of watches with glow in the dark radium paint. And they had told them that the best way to get a nice fine point on the paintbrush was to lick it with put it in your lips, called lip pointing. Pull it out like that, so you had a nice thin, and some of them had their whole faces just completely dissolved. Yeah.
Or the story about the summer camp of girls outside the Alamogordo nuclear bomb test.
Oh, did they have problems too? Yep.
The water got infected with all the stuff, and they thought it was snow, but it was just the nuclear.
Oh, I've seen that. Yes, they
Yeah.
thought it was snow. It was not snow. They all were. It was
Yeah.
snowing in the summer. No, it was not snowing in the summer. Yeah. So you've also got some infectious agents. For example, HPV. What does HPV cause? Does anybody know what cancer? It's warts, it's genital warts. Do you know what cancer you get from HPV? Cervical cancer. This is one of those that someone can pass to you. So in men, it just causes genital warts. In women, it causes genital warts that lead to cervical cancer, which is what Gardasil is. If anybody got that shot, that's what it's for, is to prevent that. So some people don't get it. Some people do get it. So, however, without mutations, organisms cannot evolve because mutations are a source of genetic variability in species. And as we talked about, some cancers are going to be a result of certain mutagens. All right, and then the condition in which an organism has extra sets of chromosomes is called polyploidy or polyploidy. What's an example of that where you have an extra chromosome? Yeah, there you go. So let's look at mutagens for a minute. Mutations. Is it going to be Granger? Is it going to be a bank? What do we think? I don't know what that is. Oh, it's a pesticide. Oh, that's a great commercial. Get the grubs out of your lawn. Yeah, that's absolutely something that can cause gene mutations. We actually said pesticides, and there's a commercial for it.
Since I was a kid, I have been curious about mutations. I think certain movies may have had an impact. I used to imagine mutations with amazing abilities, but there was also a lot I didn't understand about mutations. Let me explain. Many people understand that a mutation is a change of genetic material, more specifically, a change within a nucleic acid. RNA and DNA are both types of nucleic acids. Therefore, anything with RNA or DNA can have a mutation. That means animals, and that definitely includes humans, also plants, fungi, protists, bacteria, archaea, they can all have mutations. Additionally, so can viruses. Many mutations can be neutral in effect. You can see this codon codes for the amino acid leucine. But if it experiences this particular silent mutation, even though a base has been altered, it still codes for leucine. It did not change the amino acid. Mutations can also be harmful or helpful, but it's important to understand that mutations are random. The organism can't will itself to get a certain mutation. You can learn more about this in our natural selection video, where we talk about how a bacterium may already possess a helpful mutation that allows it to survive an antibiotic. But the bacterium didn't will itself to mutate to develop this certain mutation. Okay, so again, mutations are random, but there can be factors that make mutations more likely to occur. External factors like certain types of chemicals or excessive radiation, or internal factors like an event that causes a problem during DNA replication in interphase. During interphase, cells can replicate DNA before they divide. So let's discuss some different types of mutations. We'll start with gene mutations. DNA makes up genes, and genes can code for proteins that influence different traits. So when a mutation in DNA happens, which specifically means a change in one or more DNA bases, then different proteins can be produced, which can affect an organism's traits. In this example, we have fruit fly DNA. Mutations could include substitution, which means the wrong base is matched. Insertion, which means an extra base or bases are added in. There's also deletion, which means a base is removed. Insertions and deletions have the potential to be especially dangerous. Why? Remember in protein synthesis how we talked about how bases are read in threes? Well, if you add a base or remove a base, suddenly the number of bases total has changed. And if you read the bases in threes, depending on where it happened, everything that is read afterwards could be affected. We call this a frame shift mutation. If you look at this frame shift example here, where a single base has been inserted, you can see how the following codons, which have 3 bases each, are now all affected as the reading frame has been shifted. This example shows how it can lead to many amino acid changes. There are also chromosomal mutations. Remember that chromosomes are made up of DNA and protein, highly organized, and they have lots of genes on them. The human chromosome number is 46, with 23 from an egg cell and 23 from a sperm cell. This type of fruit fly here, however, has 8 chromosomes, so 4 came from an egg cell and 4 came from a sperm cell. Some examples of chromosomal mutations include duplication, where extra copies of genes are generated, deletion, where some of the genetic material breaks off, inversion, when a broken chromosome segment gets inversed, which means reversed, and put back on the chromosome, or translocation, when a fragment from one chromosome breaks off and attaches to another chromosome. There's more mutations than what we covered, of course, but the idea is that there are many different kinds of changes that can happen. If a mutation is going to happen, we already mentioned that there are especially vulnerable times, such as during DNA replication, but also there are other times too, like during meiosis. In fruit flies and other animals, meiosis makes sperm and egg cells that can have the number of chromosomes is the organism. However, sometimes those chromosomes don't separate completely. We call this non-disjunction. This can result in an egg or sperm cell that has too many or too few chromosomes. It's possible for a mutation to be passed down to offspring. Consider a protist with a mutation. Many protists reproduce asexually, and when they divide, the daughter cell can inherit the same mutation. A fruit fly, which reproduces sexually, can pass a mutation to its offspring if that mutation is found in the genetic material of the sperm or egg cell. You may wonder why we've been mentioning fruit flies so much. Well, they're awesome, but also it turns out that fruit fly mutations and how they are inherited are frequently studied. We have some links in the video description for more info. These studied mutations can also occur in humans, and we'll get a gene mutation example of a substitution in the case of sickle cell anemia. First, a little background. Hemoglobin is a protein in your red blood cells that helps you carry oxygen. But in the disorder sickle cell anemia, the gene that codes for hemoglobin is mutated. If you inherit 2 copies of this gene, one from each parent, you can have this disorder. This disorder can make it difficult for your red blood cells to carry oxygen because the shape of the red blood cell is affected from this mutated hemoglobin protein. This can lead to anemia and other problems. Well, unfortunately, there is not a cure for this disorder yet. The good news is that treatment for this disorder has greatly improved. Another thing to mention, if an individual only inherits 1 copy of the mutated gene from one parent, they are a carrier, but they don't officially have the disease. Usually they do not have symptoms, but those that are carriers appear to have a protective factor against malaria. Malaria is a disease caused by a protist that can be transmitted by mosquitoes. These individuals can still get malaria, but their symptoms are often less severe. We should mention that studying mutations and genetic disorders is a large and important field right now. Genetic counselors work to help families that may be affected by genetic disorders. If you have an interest in learning more about careers related to this topic, please check out some of our further.
There you go. Get the idea. So yep, lots of options for mutations, and we will talk more about sickle cell anemia in the next couple of weeks when we start talking about different genetic disorders. All right, so gene regulation and expression. What controls the development of cells and tissues in multicellular organisms? These are called master control genes, and they're like switches that trigger particular patterns of development and differentiation in cells and tissues. Now, this is actually super fascinating. So, regulating gene expression is especially important in shaping the way multicellular organisms development. So, how do you go from a single zygote, an egg that's just been fertilized? Everybody was once a single cell zygote. How do you go from that into an organism full of specialized tissues? We know that differentiation has to occur, and you have to start with a cell that is totipotent, right, to get there. But how does the cell know what to become? And this is all gene regulation. So this is actually super duper fascinating. So this is how humans look at various stages. So at one point, about 4 days after being fertilized, we all look like that. Okay? It's called a morula. That's your little ball of cells, literally just a little ball of cells at 4 days. At 4 days, by the way, can a woman typically even tell if she's pregnant at 4 days? No. So 6 days later, now you have what's called a blastocyst. So now it is hollowed out, made like a ball with a lot of cells collected into one side. Those cells that are collected into one side are going to further differentiate in the blastocyst stage. So early blastocyst, late. Within 4 days, they're going to start to differentiate. Now you see you have 3 different kinds of material there. You've got a blue, like a yellow, and a pink, right? And by gastrulus stage, you get this embryo going on here with 3 germ layers. They're called germ layers because each of those germ layers gives rise or germinates another type of cell. And over time, you get development of different types of body tissues from each of those germ layers. So you've got an ectoderm on the outside, a mesoderm, and then an endoderm, which would be the blue part there. And this would be looking at it from different angles, but we all looked like this at one point. We all had cells just organized into different areas microscopically, and then they start differentiating into other things, into other types of tissues. And there are some disorders that, like if the mesoderm doesn't form correctly, you can get some very major health problems. Yeah, it's interesting. So then we have what are called homeotic genes. So homeotic genes are any group of genes that control what kind of pattern do they control? Development and body formation during early embryonic development of organisms. So what is a DNA sequence? Around 180 base pairs found within genes that is involved in a regulation of patterns. Caitlin? Do you know? You're just like a... Yes, homeobox genes. This is a fun word. Homeobox genes. All right. So they are sequences within genes that tell that gene, this is how we're going to develop anatomically. And that would include things like animals, fungi, plants, single cell eukaryotes that have different body structures and whatever. It tells you how these things are going to develop. Like what kind of material are we going to be? And then we have these other kinds of genes, which are a subset of homeobox. Hox genes. Okay. So we have homeotic genes, and they are divided into homeobox and Hox genes. And Hox genes are a subset of the homeobox genes and are a group of related genes that specify regions of a body plan, okay, of an embryo along a head to tail axis. Actually, head to tail is probably not the best description. It can be, but when you get into developmental biology and embryology, you'll often hear about alimentary canal development being one of the first and most important derivations of cells. And that is literally mouth to anus. So your alimentary canal is everything for your digestive system, mouth to anus. And that is one of the first things that develops. And it develops in a specific order from a specific type of tissue turned on by a certain gene. So if you look here, these Hox genes, it's showing where these different genes are located. These will become head. These will become thorax. These will become tail end of abdomen. And these are fruit flies. They mentioned fruit flies. Drosophila, and that's what you're dealing with, Drosophila fruit flies. If you ever spend any time in a biology lab building in college, prepare for them to land on you and on your desk and on your notes, because they get everywhere and you use them for everything because they're very easy to work with, but they also go all over. I still remember the smell of their little nutrient base that they live on. I'll never forget it. Yeah, they're great because you can very easily breed. They come with white eyes or red eyes, and there's like certain characteristics that are very easy to tell, and you can mess with them. They're fun. But so here's your homeotic genes, Hox genes in a fly. Every gene, these are genes here. These represent your homeotic genes, your Hox genes. And if you look here, you can see what those genes express. So this one here is going to be responsible for these little mouth proboscis in the fly. That's its job. That's its whole job. I'm going to build this. And this one here, I'm going to build this joint and this leg. That's its whole job. So it's really cool that we have genes to do that. And this just talks about, this little bit over here just talks about why Drosophila are particularly useful fruit flies. Not your friends in your house, but definitely your friends in your lab. So it's from this guy, Lewis, this dude, Lewis. It became clear that a set of master control genes, known as homeotic genes, regulate organs and develop specific body parts. So what involves a series of transformations from one life stage to another? You probably learned this in kindergarten. What's it called when an organism goes through various stages of life? They look like one thing. There you go. Yeah, so metamorphosis. All right. Oh gosh, we're going back, way back, aren't we? This is kindergarten? Oh yeah, look at that. You have an egg and then a larva and then a pupa and then an adult. So there are genes that control these factors too. And some of these genes are affected by environment. Definitely temperature is going to matter here. Temperature is going to matter here. If you rear this particular breed of rabbit at less than 20 degrees Celsius, it will get black points. If you rear it at over 30 degrees Celsius, it will always have white points. Always. We talked about some turtles. You know, their eggs, depending on what temperature their eggs incubate, it can control whether you get male or female offspring. So these are all different factors. You have environmental factors, you have genetic factors, and sometimes when these things combine, you can end up with a result. For example, asthma is typically a good example of something that has both genetic and environmental factors. You tend to be genetically predisposed, and then you get certain environmental factors that then trigger the disease. And then hydrangeas, y'all know these. We have lots of these around here. Did you know that the acidity of the soil affects the color of the flower? And it's also something that is genetically controlled. So gene regulation. Hello. Maybe.
Yeah.
Maybe. Woo, she's excited about her tools, but
So you guys already know that it took me a while to get this plan build started. And to
Good
be
for
honest,
you.
even though some of it was a lumber issue,
We're
some of
excited.
it was also because I was
Have you ever thought about what a disaster it would be if the cells in your eyes started producing the same hydrochloric acid that is made by your stomach cells? Your stomach cells produce hydrochloric acid to help break down food, but you definitely don't want that in your eyes.
no.
Thank goodness that doesn't happen. But it's surprising because both your eye cells and your stomach cells contain all of your DNA. Your DNA, all of it, is found in your body cells. But see, the portions that are used need to be regulated somehow. Otherwise, we could end up with something ridiculous like eye cells producing hydrochloric acid. And that wouldn't just be a waste of resources, that would actually be very difficult to explain to your friends. You want genes to be regulated, controlled. Remember that genes are made up of DNA. DNA is used to give instructions for the production of proteins in the process of protein synthesis. But an important concept is that there needs to be a method of determining which genes will be turned on and which genes will be turned off. This is called gene regulation. And there are many ways that genes are regulated. In your human body cells, you can have proteins that can bind to certain gene regions to increase the rate of transcription for the transcription enzyme RNA polymerase. Or you can have proteins decrease transcription to the point that it may not be transcribed at all. This is a form of gene regulation. Your eye cells don't use the portion of DNA that codes for making hydrochloric acid like your stomach cells do, because there is regulation in your cells to determine which portions of the DNA is used. But we want to shift gears now to talk about a very interesting way of regulating genes that can be sometimes a little challenging to visualize. This particular method is really found in prokaryotes. a few eukaryote exceptions. It's called an operon. An operon is a fancy way of regulating genes, and it's usually made up of a few genes that can involve enzymes. Remember that enzymes are proteins with the ability to break down or build up the substances that they act on. Now let's talk about some key players in an operon so that we can see some gene regulation here. First, RNA polymerase. It's a builder enzyme actually, because RNA polymerase is an enzyme. In biology, a lot of enzymes end in that ASE. RNA polymerase is needed in order to start transcription. Remember that transcription and translation are steps in protein synthesis. Protein synthesis means to make proteins, enzymes in this case. The thing about RNA polymerase, though, it gets a little confusing for RNA polymerase without somewhere to bind. If you watched our DNA replication video, you learned about DNA polymerase and how it had to have a primer to know where to start. Well, RNA polymerase needs a promoter. A promoter is a sequence of DNA where the RNA polymerase can bind. So you would think, okay, you got your RNA polymerase, you attach it to a promoter, and boom, you make your mRNA, which eventually will be used to make a protein. But there's this other sequence of DNA called an operator. The operator is a part of the DNA where something called a repressor can bind. The repressor, if bound to the operator, blocks RNA polymerase. And poor RNA polymerase cannot move forward and no mRNA can be made, therefore no proteins. So let's take a look at our setup here. This is an example called a lac operon. Notice there's a promoter region of the DNA, the operator region of the DNA, and there are 3 genes here that code for enzymes that help in the process of breaking down lactose. Lactose is a sugar. If lactose sugar is around, bacteria want these enzymes to be made so that they can use them to break down the lactose sugar. Then they can metabolize it. Fed bacteria are happy bacteria. Here's the repressor. There's actually a gene here that codes for producing the repressor. See this gene that we call I? It has its own promoter. Well, this gene codes for the production of the repressor. So why do we need this repressor? Well, it's wasteful to make things that you don't need. If there's no lactose, it wouldn't make sense to start making enzymes that work together to break down lactose. It would be a waste. The enzymes would just sit there. So if lactose is not present, then the repressor binds to the operator, and this blocks RNA polymerase. So mRNA cannot be made, and therefore the proteins, the enzymes in this case, cannot be made. But if lactose is around in the environment, something pretty cool happens. The lactose, remember that's the sugar, binds to the repressor. This changes the repressor's conformation. Try as it might, the repressor cannot bind to the operator. RNA polymerase finds its promoter, binds, and transcribes to make the mRNA from the genes on the operon. That mRNA will be used to make enzymes to break down the lactose sugar. And bacteria like to eat, so that makes them pretty happy. We have to say that we think it's pretty impressive to think about all the gene regulation that occurs in cells. And if you find it fascinating, know that there are careers that focus on gene regulation and understanding how genes can be turned on and turned off. Because by doing that, we can gain a better understanding of a variety of diseases that have gene influences in the human body. Well, that's it for the
There you
Unique
go.
Sisters, and we remind you to stay.
No, next, no, next. Don't see you. There you go. All right, so let's look here. What are the words along the outside of the circle, Claire? Amino acids. What can you find by reading this diagram from the inside out? Caitlin.
The amino acid that binds to a specific 3-letter code.
Yeah, the codons for the amino acids. For which amino acid is AAA a codon? Jordan. There we go. Lysine. What is the code for tryptophan? Ah, Isabella. There you go. UGG. Does anybody know what's like the most commonly discussed place where we get tryptophan? You get tryptophan in your Thanksgiving turkey, and people swear it's what makes you tired after you eat a Thanksgiving dinner. It's really kind of a combination of tryptophan and mostly that you've now spiked your blood sugar and your body has compensated by producing a crap ton of insulin, and now you're going to be very sleepy. For which amino acid is GGA a codon? Drew? Glycine. Yep, glycine. What is the codon for alanine? Isabella again. Go. GCC, I think. Isn't it GC? Yeah, GCA would be good. GCC, there's a lot of them actually. Yeah. GCA, GCC, GCU, GCG. So if you have any of those, you're good. What are 3 other codons for alanine? Depending on how you did it, we'll put those up. So kind of depends in which order you did them, right? All right, so let's look over here. The change of one base to another in a DNA sequence is a substitution. There we go. B, substitution. A change in one or a few nucleotides that occur at a single point in a DNA sequence is a again. A C, point mutation. Part of one chromosome breaks off and attaches to another. Jordan. It's location. F, a heritable change in genetic information. Caitlin?
Mutation.
I'm gonna stuck that in the wrong one. Yes, A, mutation. A mutation that produces an extra copy of all or part of a chromosome. Drew? 5, H, duplication. 6, a chromosomal mutation that reverses the direction of parts of a chromosome. Colin? Inversion. G, a kind of mutation that can change every amino acid that follows the point mutation. Jordan? There you go, a frame shift. That's D. Very good. The addition of a base to the DNA sequence is a Eunice.
Insertion.
An insertion. That's number 8. Why don't you click E? Yep. All right, so a substitution, the description would be one base changes to a different base, and the outcome would be that it usually affects no more than a single amino acid and sometimes has no effect at all. An insertion is when you add a base to the sequence, and the effects can be dramatic because it causes a frame shift. It shifts the entire series. A deletion is when you remove a base from the sequence, and its effects can be dramatic because it causes a frame shift and shifts the entire series. Deletion can happen as a gene mutation or as a chromosomal mutation. What's the difference, Caitlin?
Gene mutations involve changes in one or a few nucleotides. Chromosomal mutations involve changes in the number or structure of the chromosome.
That's exactly right. Right. Perfect answer. So gene mutations, deletion happens when a base is removed from a DNA sequence. In a chromosomal mutation, a deletion involves the loss of all or part of a chromosome. 16, an embryo develops different sets of genes regulated by Caitlin.
Transcription factors.
C, very good. The process through which cells become specialized in structure and function is Eunice. Oop, that was out of order. Oh, we'll have to come back to that. Yeah, okay, yeah. 18 would be B. Homeotic genes are master control genes that regulate organs that develop in specific parts of the body. And then 19 would be, what role do homeobox genes play in differentiation? A, they code for transcription factors that activate other genes important in cell development and differentiation. 20, in flies, a group of homeobox genes that determine and identify each segment of the fly's body are a group known as, Drew?
D,
Hox genes, D, right. Clusters of Hox genes are found in what? What do we have? I drew again. There you go. D, nearly all animals. Yep. The switches that trigger particular patterns of development and differentiation in cells and tissues are called, Eunice?
Master control gene.
Yes, B, master control genes. And 23, metamorphosis is what, Caitlin?
Series of transformations.
There you go. Yes. All right. And then obviously C, which we did not have. All right. So you did this for homework. Somebody, one of you, tell me one interesting thing. One interesting thing that stuck out to you. Yeah. Yeah. I just asked, but somebody tell me something that you remember. Drew, you tell me. They're what? that DNA strands are 8 atoms wide. That's okay. That's pretty fascinating. All right, very good. So we got one thing there. All right, this I'll just play in the background. It's kind of interesting. This is Lewis, the guy that discovered the Hox genes. He has a bunch of witty quotes. I'll play it quietly in the background. You have about 25 minutes to work on your cold case. So go on back to it. I'll put those out on the counter. So yeah, if you want. I'll move these over here. Here, get this last one out. Yeah, let me put these over here where people can get to them. Answering all the questions. I would. Why not?
Yeah, I have the column.
Yeah. I didn't pay out. I actually go as well, actually, we probably pay alcohol later. Yeah, I think it's why you didn't know like just get all the your friends that you have access to all because you know all these effective I mean, all we have to do is like food. I mean, the timeline should is great. Either way, we should probably stop. The timeline should have more than less than. We should be able to get it off the timeline. I do advise that we switch aims. No, so you said including strategy. That's what also. Yeah. But in case we can. I'm lying. That's the
Somebody ate some pizza.
I'm really proud of you buy what a green for your family buy. Thanks.
Well, I think that's a good quote.
Yeah, he does. The real good one. It looks like Elon Musk's angry brother right there. We're doing a lot. I know. I'm not sure why.
Our goal is to try to leave. It does. Our.
People are the bad things that you just I agree.
I think we're just supposed to keep going until we finish. It's ever been.
Are we allowed to ask for more than one person's fingerprints?
Yeah, you're gonna ask for a lot of people's fingerprints, but one in particular you're looking at as a suspect, and the others, or at least one, and the others you're looking at as.
So is there anything stopping us from just getting all of them?
Well,
Public's gonna approve a warrant fingerprints, especially if you've never been in that department. But that's an overreach of the state. So you have to have a reason for this, because it's, who would have been in that house? Like, is there any reason to think that, you know, the paperwork was inside of it? No, they touched the paper, does that matter? Gotta find other people.
One by one, how those people look back at each other.
How they're related to your unsub.
To the guy, the Irish guy.
Yeah, your unsub is related to all three people. So you have to find where these people disagree with us, that's what you say. The unsub is, that's your unknown subject. That's whose DNA this is. You need to follow these family trees. And everybody is in there better than I was.
Yeah. I wonder if these have. I don't think they have. Yes. Okay, so one by one, the penguins, well, probably have, well, big, and June big, they've probably been here before. Actually, you should get like a separate piece of paper to make name of this.
Oh, it's right here.
Sure. Okay, so Bob Blue and June Fake